All right, y'all, welcome into the Meiosis chromosome bead demo. Special shout out to Carly Dollar, who is a Biology 1021 GTA, who you will see shortly in this video. We recommend that as you're watching, that you draw out a diagram of what's happening so you don't have to go back and watch the video again. So first, you are going to separate the little beads based on color and size. So at this point, you should have 2 sets of red chromosomes and 2 sets of yellow chromosomes. The second step is you're going to take 1 chromatid away from each set. So at this point, you should have 1 chromatid of each color and each length on your tray. So in this phase is called G1. At this point, the cell is just chilling out, hanging out. It's getting ready for the next phase. And the next phase here is the S phase. So the S phase, S stands for synthesis. During this phase, the chromatids, they are going to replicate, so we're simulating this, by putting all of the matching sizes and colors back with their friends, and we're grouping them together in a little ball like you're seeing here in this video. So the next phase is the G2 phase, and basically this is where the cell is getting bigger, it's growing more, and it is getting ready for the next phase. Now, I want to make a quick little disclaimer here. All of the 3 steps that we just talked about, so G1, S, and G2, are all part of interphase, as you can see here in the cell cycle diagram. Now, interphase is where the DNA is present as chromatin, and chromatin is an uncondensed version of DNA. Now, interphase is not a true part of mitosis or meiosis, so we are about to start discussing the steps of meiosis. We'll begin talking about meiosis 1, and then we will finish the video by talking about meiosis 2, because there are 2 cell divisions that occur during meiosis. Okay, so this part of the demo represents prophase 1 as part of meiosis 1. P, you can remember that as prepare. So what you're going to do with the beads is that you are going to put them into 2 groups. You're going to put all of the short ones together, and then you're going to put all of the long ones together. Okay, so some important things to note about prophase 1 during meiosis 1 is that crossing over occurs. So the chromosomes are condensing, spindles are forming, and the nuclear envelope starts to disintegrate. But the biggest thing we want you to remember about prophase 1 is that crossing over occurs. So what you are going to do with these beads, you're going to start taking some of these beads off and trading them with other sets of chromosomes, like you can see here in this video. And it's completely random. Crossing over is completely random, so it doesn't matter how many beads you do for each set, it does not matter. Okay, so the next phase is metaphase 1. So the tetrads are lining up in the middle of the cell midplane. Okay, so the next step is anaphase 1. A you can remember as apart. During anaphase 1, those homologous chromosomes are going to separate on either side of the cell. And as you can see, Carly pointed out just a second ago that she forgot to do crossing over on the longer sets of chromosomes, but that's totally okay because it's completely random. The next step is telophase 1, and during telophase 1, the nuclear envelope starts to reform as now there are 2 cells, okay? So cytokinesis has occurred. So now we've got 2 cells on either side of our tray. So now that we're beginning meiosis 2, we're starting off with prophase 2. So the cell is simply going to prepare itself again. That nuclear envelope is slowly going to disintegrate. And now we're going to move into the next phase, which is metaphase 2. So for the sake of simplicity, remember we've got 2 cells, but we're just going to simulate 1 because that's all the room we have here. So the chromosomes, they're going to line up in the cell midplane. All right, so our next step is anaphase 2, where the chromosomes are going to separate apart into their corresponding chromatids on either side of the cell. And the last step of meiosis 2 is telophase 2. And during this step, the chromosomes, they're going to gather at the two poles of the cell. And the cell is going to divide via cytokinesis. A cleavage furrow is going to form in the case of an animal cell. And this is going to form two daughter cells. And the ploidy is going to be haploid, abbreviated as the letter N. Now remember, I just mentioned a second ago that two haploid cells formed as the result of one cell going under meiosis 2. But remember, we had two cells. So grand total, we are going to form 4 haploid cells, which is what this diagram here is showing on the far right. So this whole entire diagram is showing a complete summary of meiosis 2. I hope that this video was helpful and kind of reminded y'all of the simulation that you did in lab this week. If you have any questions about this video, please feel free to email me, Madeline, or your lead GTA.